about 63 kilodaltons. Now, this indicated that PFRH5 existed as a higher protein complex. In order to have a proper control for this, since Alan Coleman has already shown that RIPAL is involved in the interaction, we had to make PF-RIPAL the exact region Alan Coleman had made. Now, PFRH5 here, I already mentioned that it migrates at higher protein complex. So using RH5 antibody, we conducted immunoprecipitation experiments where we eventually found that these antibodies against RH5 did immunoprecipitate RIPAR, consistent with Alan Coleman's report, immunoprecipitated RH5, and the third interacting protein, SIRPA, which we initially started studying. Now, we took the co-immunoprecipitated elutes and analyzed them by immunoblotting and found that antibodies against the interacting protein detected the corresponding proteins in the co-immunoprecipitated elutes, whereas the pre-immune sera did not detect any of these proteins in the samples. But then, using an unrelated control, which is PFRH2, our antibodies in fact detected RH2 in the lysate, but not in the co-immunoprecipitated elute. This, to some extent, tells about the specificity of our antibodies. Now, we try to localize SERPA, given that this group in Switzerland has localized this protein to, they've reported that it's co-localized with MSP5. MSP5, nobody knows where it is localized, actually, because it's sparsely characterized. Now, using micronemal resident antibodies against proteins, we found that SERPA co-localizes with EBA175 as well as PF-RIPAR, suggestive of a micronemal location. We did analyze this parasite called schizone lysate by anion exchange chromatography, where we're exploiting the isoelectric point of these proteins, and found that even though by step gradient elution we had 3 peaks, we detected the 3 interacting proteins in peak 1 using the 3 different antibodies. Now, looking at figures B and E, we proceeded by analyzing schizone state lysate by size exclusion chromatography. Our point here is that interacting proteins should co-elute in the same fractions. Upon analysis by SEC, we detect, using our antibodies against the interacting protein, we detected under denaturing conditions the respective interacting protein, both with parasite schizone lysate and culture supernatant. Now, when you analyze schizone lysate and culture supernatant fractions by blue native page electrophoresis, you find that they all coexisted as single protein band, which reflects that these 3 proteins are in fact interacting and they form a higher protein complex. Now, PFRH5 SERPA complex, we feel is GPI anchored. Now, what happens when you conduct 3D reconstruction of IFE-Z stacks? You find that invading merozoites, which have been treated with cytochalasin D, and you do the staining, RH5 is found at the apical pole, whereas you have MSP surrounded, and then the nucleus of the parasite stained by DAPI. Similarly, when you do that with RIPAR, and then with CYRPA, an indication that these 3 interacting proteins are in fact on the surface of the merozoite during erythrocyte invasion. We tried to conduct Triton X114 phase partitioning experiment to see if SERPA is membrane associated. And of course, we found SERPA in the detergent fraction as compared to PFRH5, which we found in the aqueous fraction. We tried to look at the omega site of previously reported GPI anchored proteins, and we found that SERPA contains the omega site consistent with previously reported GPI anchored proteins. But since RIPAR is not GPI anchored, it doesn't have the omega site. We did tritron radio labeling of SERPA in schizone state parasites that has been stained with GPI moieties. After co-immunoprecipitation, we got a signal indicative of SERPA. Although one would expect that there should be some unlabeled SERPA as well as the other 2 interacting proteins, which we use our antibodies and in fact detected them. Now, antibodies against the partial construct of SIRPA and RIPAR poorly inhibited erythrocyte invasion, whereas the full length of SIRPA in both 1 and 2 cycle invasion assays, just as efficient as PFRH5, did inhibited erythrocyte invasion. Now we took antibodies against the full length and tested against both sialic acid dependent and independent parasite lines, and we found that SIRPA has cross-strain transcending invasion inhibitory activity, just as been reported for PFRH5. Thank you. When we did combination of SIRPA and RH5, we found synergistic invasion inhibitory activity both in sialic acid independent and dependent parasite line. We got the greatest shock 2 weeks back when Alan Commons group published another paper in Cell Host Microbe, where he claimed that this interaction is in fact required for erythrocyte invasion, but that SIRPA is not GPI anchored. Now, my opinion about this report, even though he has linked SIRPA to play a significant role in cation signaling downstream of erythrocyte invasion, my point is that he has reported that he conducted the same tritron radiolabeling experiment and it was unsuccessful. I didn't see any blot and there was no positive control. And in the absence of that and no experiment he has shown where they have attempted to mutate the omega site and assess viability of these parasites. In the current absence of that, I feel we could still position by saying that in conclusion, we have SIRPA, which is GPI linked, that secures PFRH5 and RIPAR on the merozoite surface during erythrocyte invasion. I wish to acknowledge my supervisors way back at the ICGB in New Delhi, the doctoral committee, and among others, the instrumentation facility heads, and for WACBIP also, that's provided me the opportunity to look at novel invasion-related proteins and exploit them for vaccine development. Thank you for your attention.
Well, he says he disagrees with people who are published. So if you disagree with him, it doesn't really matter. Okay, so we'll take a couple of questions. I see.
Yes.
Yeah. I don't know your name, so you can mention.
My name is Aniwa Yao, and I also work on an erythrocyte invasion.
Good.
My question is, SIRPA is in the microname.
Yes.
RH5 is in the rupture name.
Yeah.
So at what point in time do you think this complex is formed?
Good. Thank you for that question. Fortunately, you are coming from a lab where this disagreement started. Actually, Chetan Chitnis believes that the first environment that these merozoites come in contact with is of low potassium. And low potassium triggers calcium spike, okay, that triggers the release of micronemal proteins first, followed by rupture discharge. But your PI is saying that monoclonal antibodies against RH1, all right, actually blocks calcium regulation, and in fact blocks the secretion of EBA175 from the microrim, and in turn has an effect on tight junction formation. I don't know what to agree with at this point, but my opinion is that this complex is formed even at the Schizon stage of the parasite development, because when we try to assess parasite culture supernatant, we found the complex. If you take Schizon state parasite lysate, we found the complex. And of course, since processing event is occurring during Schizon state development of the parasite, of course, we could in fact see both the full-length parasite protein as well as the processed fragment. Okay?
There was a second hand.
My name is Selassie Telechu. Thank you for the presentation.
Yes.
Well, I just wanted to ask if you controlled for differential expression of the various proteins in your experiments.
Differential. Yeah, we had NAPOL. We had NAPOL as a loading control that tells us about that.
Okay.
Yes.
Okay. Because I didn't see that in any of.
Oh, I see.
So your disagreements can go on. Maybe I suggest you work in the same lab and then you generate results and agree from there. Thank you very much, Emmanuel. Let's give him a applause. Well, we will take the third presentation. Is if we are around? Okay.
Good afternoon. My name is Joelna, and thank you for giving me the opportunity to talk about this little insect that has caused a lot of problems globally. I'd like to talk about work that I did as a postdoctoral fellow here at Noguchi, and which has informed what I'll be doing also as a WACBIB fellow in the next few years. The midgut of the mosquito is a very hostile environment for Plasmodium parasites. There's a lot of bottleneck in here, and eventually parasites probably never become eukaryotes or oocytes and never become sporozoites for them to be transmitted. This hostile environment is also heavily inhabited by bacteria, which the mosquito uses for its own homeostasis and other physiological functions. What is most interesting to the scientific world about the midgut is that bacteria in the midgut has been found to have Plasmodium killing characters. So bacteria is being, thank you. The bacteria in the midgut is further being investigated for potential biocontrol strategies or Plasmodium blocking strategies. However, most of the work that have been done on this have used laboratory populations. Situations like this could happen if we don't really look at natural populations, especially in malaria endemic communities or countries, because the native microbiome of the mosquito resident in a malaria endemic country could impede the progress of any introduced bacteria at all. Apart from that, natural populations itself could have their own naturally occurring symbionts, which could be more useful to this transmission blocking property. So they need to be looked at, natural vector populations in endemic communities or endemic countries. And this have shown in two papers here, which have actually isolated Enterobacter species, the first one in Anopheles gambiae in Zambia, and Chromobacterium in Aedes, which have actually gone on to be effective against malaria and dengue in these two vectors or mosquitoes. Therefore, I find it not only important to have a closer look at natural vector populations, but also to find the different factors that could influence the bacterial composition in the natural population. One of them is the larval environment. We have the breeding sites, so how are the larval environments dictating or influencing the bacteria that is in the vectors that we have? Two is the feeding regime. Does sugar or blood meal change any of this bacterial composition? Because at the end of the day, the parasites are transmitted after a blood meal or through a blood meal. And also recently, antibiotics in ingested blood have been shown to reduce bacteria diversity and have actually reduced vector competence. So all these three things, all these three factors, we need to be looked at in the natural vector population. So I'm setting out to do something more, look into what we have here in Ghana, which is it is a malaria endemic country, we all know that. So I started by first cataloging the gut microbiota in Anopheles sampled in breeding sites in Ghana, and then looking at some of these factors, ecological factors, and biological factors that could affect the bacterial composition. And later on, look at how these can affect the susceptibility of Anopheles gambiae. So the methods were quite simple. I had my two sites in Accra, down south, and then Kintampo in the Brongahaf region, in the middle of Ghana. In Accra, well, it's largely urban. I collected my samples from very close to around the airport area. And then in Kintampo, in villages which were on a line going up towards the north. So these are some of the sites from where I collected my mosquitoes from. And largely, I collected them as larvae or pupae, mostly pupae because pupae have finished feeding. They've collected all the, have stopped feeding. They've collected all the bacteria that they would collect in their, as they fed as larvae. And then I brought them to the lab, waited for them to emerge as adults. And for the microbiota catalog, I used unfed female mosquitoes. And for the other experiments, I either fed them on sugar for 3 days or blood fed them and then dissected the midguts after 24 hours. I amplified the 16S rDNA of bacteria and then analyzed my analyzed the DNA by parasequencing. So first, I wanted to look at how many bacteria families we had in our mosquitoes. And what is clear here is that we typically have very low bacteria families, usually between 5 and 8, not very many at all. And I wanted to see how these varied between the variables that I was looking at. So one was to look at dry and rainy season. I actually collected my samples in the dry and rainy season to compare. And you could see that here, there is significantly high bacteria families in the dry season, the mosquitoes collected in the dry season, and also in the urban areas. The two species of mosquitoes that I looked at in Anopheles gambiae SS, which is Anopheles gambiae, formerly known as the S form, and Anopheles gambiae coliti, formerly known as M form, were very similar. So I went further to look at which families were there anyway. And so here there's a plot of average bacteria proportion on the left axis and on the on the y-axis, and on the x-axis I'm showing here the sites from which the mosquitoes were sampled from. So every different color here shows a different bacteria family, and here I plotted families that were more than 1% in any bacteria in any midgut. So here you can clearly see one color that is dominating in both seasons, dry season and rainy season. This yellow, I don't know whether you see it as yellow or green, but it's green. So this family is actually Shewinelaceae, which had only been isolated from Anopheles defensi in Iran, but quite recently also in Burkina Faso. I think it's just, it was published this year. So you could also see here that the more colors you have, it means the more bacteria family you have. So I wanted to see, I was observing here that the dry season had a bit more color, a lot more colors. Look at this site right here, a lot more colors, rather than the rainy season. So I wanted to see whether the diversity was significant at all or anything important. And that was true. We used the Shannon-Weaver index to look at the diversity of these families between the dry and rainy season. And this points out that there was more bacteria diversity in the dry season than in the rainy season, which was quite surprising because we don't have a lot of Anopheles gambiae in the dry season. But here we are, the little that we collected has huge diversity, which could actually be important for vector competence. So for Shewinelaceae, I went on to have a closer look at how they differed between the variables, and actually it's only between rural and urban that we had any significant difference between them in terms of how many or how much the prevalence of Shewinelaceae. Species or families Enterobacteriaceae, which have known species that have been implicated in Plasmodium killing, were rather low in our samples. I went ahead now to look at what feeding would do to this bacterial composition. I used sugar and blood feeding, and for Shewinella, which is, I went down here to use the genus rather than use the family level, because that would give me a bit more in-depth. So Shewinella was largely decreased whether the mosquito was fed on sugar or blood, which suggests to me that Shewinella may not be important in digestion of food in the mosquito. It may rather be more important in development because these were unfed mosquitoes and they came right from larvae, pupae up. So they're probably not really important in feeding, in digestion of food in the mosquito. However, these two species Enterobacter and Sedecia, which are both Enterobacteriaceae, were increased significantly after blood feeding. So if somebody has isolated Enterobacter species that have shown to be important in Plasmodium killing, it could be important for us here in Ghana as well, because these Enterobacter species have been increased by blood after blood feeding. So we don't know what these species do, but it may be important to further look at what functions they actually play after blood feeding in the mosquito gut. In Ghana, I mentioned antibiotics or antimicrobials, and a study by one of our own here at Noguchi, has shown that these three antimicrobials are three of the most administered antibiotics or antimicrobials. So I wanted to have a closer look at them and see what effect they had on the bacterial composition. So I spiked blood with various concentrations of these antibiotics and then did the pyrosequencing analysis again. And here for Shigella, which is our most dominant bacteria, amoxicillin decreased. So here on the y-axis is the ratio of the bacterial proportion to the control. So anything below 1 means it was reduced as compared to the control, which is just blood fed. And then anything higher than 1 means there was an increase. So here it was only amoxicillin which reduced Shigella significantly at higher concentrations, which is 15 microgram per ml. But then for metronidazole and tetracycline, it seemed to rather go higher significantly with increased concentration. And then for Enterobacter, this was quite expected. Amoxicillin affected it at higher concentrations, and metronidazole as well, but tetracycline increased it at higher concentration. It will be interesting to look at what metronidazole actually does, because it's known to affect protozoans. So one interesting one interesting direction is to look at if a mosquito has picked up metronidazole from a host, what direct effect it would have on the Plasmodium parasite, since it's also a protozoan. So in summary, for Anopheles gambiensis in Ghana, they have very few bacterial families. Microbiota diversity is higher in the dry season, which is an interesting field that we have to look at in terms of vector competence in the dry seasons. Bacterial composition is influenced by season and locality, and for my data, it shows that dry season and urban locality actually affects it. It's dominated by Shigella. I've already mentioned this. And blood feeding increases Enterobacteriaceae, which may be a good thing depending on what functions the Enterobacter does. And then amoxicillin decreases Shigella and Enterobacter, which may not be too good because amoxicillin is the most administered antibiotic. So for my current study, this is what I intend to do as a work-based fellow. I actually really want to set up a wild laboratory population. Wild because we need large populations to do some of these work. And since I want to base my focus on field population, I want to set up a protocol that would help me maintain mosquito populations, but still keeping the natural gut microbiota. It may be difficult because once you bring mosquitoes from the field and put them in tap water, it changes completely. The microbiota composition will change completely. Isolate the bacteria I've already found, and then look at the functions in terms of development and susceptibility to Plasmodium in the mosquito. These are some future directions that I'd like to undertake. Especially of most interest to me is the anti-Plasmodium mechanisms, because we still don't know. They say it reduces the oocyst number, for example, in the presence of bacteria. But what actually happens to the Plasmodium, nobody has actually thought of doing that. So I'd like to acknowledge all these people for the various contributions they made to my work. Thank you.
Oh, you want to take the mic from me? By me? Okay. You will become a pillar of salt. So I wasn't going to give time for any, or should I consider her the first lady in my... Okay, any quick question or interaction? Okay. Wow.
Thank you, Selassie, once again. About the blood feeding increasing Enterobacteriaceae, I don't know about the nutritional needs of that family of bacteria, but could it be possible that the blood provides the nutrients they need? That is why there's an increase in the population, rather than they having a role to play in digestion or something else?
Thank you. It's actually both. So some of these, I think there's a paper that I came across, I don't remember the author, but they actually mentioned that some of these bacteria actually need the blood for growth. But like I'm saying, it's a symbiotic relation. So the mosquito, by the Enterobacter increasing, the mosquito also benefits from digestion as well, because the bacteria helps it in digesting things.
Any reasons why you left out Northern Ghana? The director says
Oh dear. Nothing. Nothing personal. It was just. No, it was just really. I wouldn't say it was random, but I wanted urban. I didn't. It wasn't to go to a highly endemic area really, but just to give some kind of geographic difference. Okay, thank you.
All right, so is it a burning one? Okay, hello.
Please, do you detect Wolbachia?
No. In the gut, these are Anopheles. We don't really expect Wolbachia. It would have been a groundbreaking result to find Wolbachia in Anopheles, but no, I didn't find any.
Okay, so is there amoxicillin reducing some bacteria?
Reducing?
Yeah.
Yes, Shawonella.
Okay, so did it increase vector competence?
Okay, so that's the thing. Because these are filled samples, you need, usually for infection studies, you need a very large number of mosquitoes. Because I was even doing laboratory infection. So that even gives you much of a bigger challenge because you need a lot of mosquitoes. But if you go to the field each time, you don't have the number of mosquitoes that you need for such a huge experiment. Again, when you're doing such experiments, it's age controlled because the older the mosquitoes get, it brings all sorts of complications to the experiment.